Good afternoon. We're here today with Ben Buelow, who's the Chief Medical Officer for TeneoBio in Menlo Park, California. And Dr. Buelow has some interesting preclinical research that he is presenting here at ASH in 2017. So tell us a little bit about your technology at TeneoBio.
Sure. Thanks for having me. So our bispecific molecule that we're using in myeloma is based on two really seminal technologies at TeneoBio. The first one is our UniRat, which is a rat that makes fully human heavy chain only antibodies. And that synergizes very well with our antibody discovery pipeline, which is next generation sequencing based. And overall, that allows us to discover a much broader array of antibodies with unique functional properties than other types of antibody discovery platforms. And what we've used that to do is essentially create a suite of T-cell engaging molecules that span a number of kind of unique functional properties in T-cell engagement. And that's really well represented by the two molecules that we are discussing at the conference tomorrow, TNB384B and TNB383B, the latter of which is a molecule that we are hoping to advance into first-in-human trials in late 2018. And what we've done in those studies is just to show that while both molecules are able to activate T cells and show really good efficacy in preclinical models of myeloma, that TNB383B appears to do that with minimal cytokine secretion. And we're hoping that that's going to limit toxicity from those molecules and also make them very amenable to combination therapy.
So there's a lot of interest in T-cell therapies in multiple myeloma and of course across a lot of different hematologic malignancies. So how does yours differentiate from what other companies are doing?
Yeah, so that's a great question. And you're right, there is a lot of interest and a lot of hope, I think, because I think we're all encouraged by the trial data coming out of the CARs and some of the antibody-based therapies as well. However, all of those therapies, the price tag, so to speak, of the efficacy is the toxicity. And as I mentioned, although we're still working on the mechanistic underpinnings of this, and I'll speak about that tomorrow at my talk, we have some idea about it, but for reasons that we're still working out, TNB383B engages T cells in a way that causes them to still lyse tumor cells. It still shows the same maximal lysis as molecules that are really strongly activating and activate all T cells, but does that without triggering as much cytokine release. And as you know, most of the toxicity from these T cell engagement therapies are mediated by uncontrolled cytokine release. So our hope is to have both a better toxicity profile and part of the problem is that because these therapies are so toxic, physicians may hesitate to combine them with other therapies that also have their own associated toxicities. So our hope is that this will also create a better opportunity for combination therapy.
So I think you said you're going to hopefully be in first-in-human trials next year in 2018?
Yeah, in late 2018. We're a small company, and that makes us very nimble. And we are well on track to getting our IND submission in late Q3 of 2018. Hopefully, getting our first-in-human trial started in late Q4 2018, maybe early 2019.
Well, that really interesting science. I wish you the best in all the work that you're going to do to get into those human trials. The population needs it. Although there are great therapies, we need more. So thanks
always
for coming by and introducing us to your technology.
Great, thank you so much.
Thank you.